the suspected samples. Mr. Blaise, please, floor yours.
So, good morning, ladies and gentlemen. And first of all, I would like to thank the organizers for allowing me to present this work about the use of lateral flow device for safe and low-cost shipment of FMDV suspected samples. And this work was mainly performed by my colleague Aurore Romet, and in the framework of a project funded by EuFMD. So, what about the context of the study? So, as you are aware, towards global control and eradication of FMDV, there is important to identify infected region, to identify circulating strains, and to study virus dynamics. But in endemic arrays, there is often a lack of FMDV laboratory diagnostic capacity, resulting in, and also low or no submission of infected samples to reference laboratories. And the major obstacle is the cost of shipments due to the biological risk, so the containment of samples, and also the conditions of transportation under shield or freeze conditions. So, that's the reason why our aim was to develop a low-cost and safe method for shipment of FMDV suspected samples based on the use of lateral flow device, developed by Svanodip and purchased from Boehringer. So, these pen sites are immunochromatographic strips that are routinely used on field for the pen site detection of FMDV antigen. The sample are vesicular fluid or lesion epithelium, and accurate result is available after 10 to 30 minutes. And the idea was to use positive LFD device for regular shipments, for shipment under less stringent conditions of FMDV suspected samples. So, for example, ambient temperature and triple packaging. But the major concern was, what is the safety of positive LFD for shipment? And to address this issue, we deposited a different load of FMDV Omanisa on the LFD, and we let the samples, the suspension migrate as routinely performed for field samples, and we observed positive, dopeful, or negative results according to the viral load. And after 30 minutes, we disassembled completely the LFD and grinded the strip in culture medium to inoculate the resulting suspension on cells. And we observe cytopathic effect within 18 to 30 hours post-inoculation. And we also disassembled another LFD and completely grinded the strips in a lysis buffer to perform RNA extraction and RT-PCR targeting the 3D coding regions. And we obtained also positive CT value. Thus, infectious virus is eluted from loaded LFD, so positive, dopeful, even negative. And we also perform other investigation, and we obtained that live virus is still recovered in 18 hours, 7 days, and 14 days after LFD loading, positive LFD being stored at room temperature for the indicated time. Thus, inactivation of LFD loaded with FMDV is required to ensure safe shipment. So, based on the literature, we choose 2 inactivating reagents, so sodium hydroxide or citric acid. And we pull viral suspension with the same volume of citric acid, and we for 15 minutes, and we inoculate cells with these suspensions. And we perform preliminary investigation to set the correct concentration range to avoid cell toxicity related to the chemical reagent. And as you can see on this table, the conclusion is that 0.2% citric acid solution completely inactivates FMDV in solution in 15 minutes. And to adjust the right contact time between the reagent and the virus, we perform different tests from 15 seconds to 15 minutes. And as you can see, 1 minute incubation with citric acid solution is sufficient to inactivate FMD strains in solution. But for biosafety issue, we decided to set the contact time at 15 minutes. And we evaluated this inactivation of FMDV in solution on a panel of 19 reference laboratory strains available in the lab and representative of the 7 serotypes of FMDV. And as previously explained, we perform inoculation of sensitive cells, 2 passages, 2 times 48 hours, and also RNA extraction for RT-PCR detection targeting the 3D. And overall, we conclude that 15-minute incubation in citric acid solution is sufficient to inactivate FMDV strains representative of the 7 serotypes, while the 3D coding region is still detected by RT-PCR. But our aim was to be able to inactivate the virus directly on the LFD. So we test a first protocol. So just to mix the virus with the commercial diluent to deposit on LFD, to allow the samples to migrate for the time recommended by the purchaser. And after 30 minutes, we add citric acid directly on the device and for 15 minutes. And the solution, so the LFD was completely disassembled and grinded in culture medium to inoculate Abarus2 cells, but it doesn't work. We observed cytopathic effect within 17 hours post-inoculation. So we evaluated another protocol. So, as previously, deposit of virus diluted in the diluent, and after reading of the results, so 30 minutes after deposit, just to soak completely the device in a citric acid bath for 15 minutes. Then, to recover the LFD, to disassemble it, and to grind the strip in culture medium to inoculate Abarus2 cells. And in this case, no CPE was observed after 2 passages. So we were quite happy with this protocol. So we move on to evaluation with reference strains. So live virus is inactivating by soaking positive LFD in citric acid solution bath during 15 minutes. So we use this protocol on a panel of 7 reference strain lab available and representatives of 7 serotypes. And we deposit the virus on LFD. We observe the results. So as you can see, we observed a positive result on the LFD. And after that, we soaked the LFD in citric acid solution or in water for control protocol. And as you can see, after elution of LFD and inoculation to sensitive cells, we observe that the virus is correctly inactivated. And we also performed RNA extraction, RT-PCR, VP1 amplification and sequencing, and we also performed genome transfection from the RNA extracted from positive LFD. And we were able to amplify the VP1 and to sequence the VP1. We were also able to detect the genome and targeting the 3D and the RS region. And we were also able to rescue the virus after RNA transfection, so chemical transfection. And after transfection, the virus were confirmed by ELISA antigen capture. So overall, after inactivation, no CPE observed. Genome is still detected by RT-PCR. VP1 is sequenced and virus rescued after RNA transfection. Since in endemic arrays the ambient temperature is usually high, we also evaluated this protocol at 37 degrees, and the modification shown is just a step, the inactivation step at 37 degrees versus room temperature. And we observe that this protocol is also effective at 37 degrees, as you can see from this table. So, we finally evaluate this protocol to field samples. So, first of all, field samples available in the laboratory, so 3 field samples. And we obtained results in line with those obtained with reference strains. And we were able to rescue the virus for 2 out of 3 reference strains. And we also evaluated this protocol on field samples sent from Kenya to ANSES. So thanks to Keith Van Manen and Dr. Sangula. They sent this LFD as positive LFD, previously soaked in citric acid. And from this positive LFD, we were able to detect. So the virus was effectively correctly inactivated. We were able to detect the genome, so targeting 3D and iris, and we were also able to sequence. The sequencing was not done. We were not able to rescue the virus, but overall, the inactivation was okay, as the genome detection for all samples and virus rescued for field samples available in the laboratory. So, based on these results, we propose a procedure from farm to reference laboratory. So, in farm, the samples could be, or on field, the samples could be collected and deposited on the LFD. Then the LFD could be soaked in citric acid. Then the citric acid could be discarded. The tube could be decontaminated. And then the LFD, positive LFD, could be sent in triple packaging at room temperature to reach the reference laboratory for further analysis. So I'm going to conclude. We have developed a protocol for low-cost and safe shipment of FMDV suspected samples. We evaluated it on reference strains and 9 field samples. And we propose a procedure for collection, then safe shipment. And the perspective of the project are to evaluate the protocol on a larger panel of field samples, to experience the procedure on field, and thus the collaborations are welcome. And it's time to acknowledge the colleagues, the colleagues on field, and also you for your attention. Thank you.
Thank you very much. Is there any question?
Okay.
Don't debate.
Yeah.
So thanks very much, very nice presentation. Thanks, very useful information. So thank you very much. Very useful presentation. And now I can speak. I suppose thinking about shipping materials from the field to the lab, I'm sort of questioning why you need to inactivate. If you're going to send them via Category A or Category B, so in your scenario here, you're shipping the material as Category B. Do you really need to inactivate all the virus on the LFD?
If you want to send the LFD or only the strips without inactivation, the risk is to have still virus on it. From these results, if you want to don't inactivate, you have just to you have to put it in lysis buffer. But for me, it's important to inactivate to be able to send safely.
But I suppose if we were going to send the samples through regular post, it would be vital, and I guess we would come up against some significant problems there with full-length RNA being still present on the LFDs, and probably the regulators would not allow us to ship material without any UN 3373 or 2900 regulations. But if we're going to send them via Category B, do we really need to do, I'm still questioning whether we really need to kill all the virus that's on the sample.
I think.
May I comment?
It is the same comment?
No, my comment to Don is, are you sure that it is a diagnostic sample, a LFD positive for FMD, or it is a virus positive for FMD? It's more...
Can I also make another comment? Is that obviously we started this work at Pirbright a while ago, and one of the key things that is different, that what we see at Pirbright to what you see, is that when we use clinical samples on LFDs, we are not able to recover live virus, which is different to what you're showing when you use cell culture supernatants. So I think something that would be very important for us to do before we start getting carried away too much is to look at if that is a real observation, because obviously we will not be shipping cell culture supernatants from countries where we'll be shipping real clinical samples. And I think there's a really nice opportunity to see whether that is a real observation, because in my experience, when we have been using clinical samples on LFDs, you cannot recover the live virus without electroporation.
Yeah, no.
So there might be something quite interesting in terms of antibody, some sort of mucosal response, which is helping us with our biosecurity in that it's limiting the ability to be able to recover live virus.
Yeah, but I think we have to collect clinical samples to be able to conclude, because as you can see, we experienced the protocol on very few samples, but anyway.
Well, yeah, so we have continued to work with real clinical samples, and in my talk next, we have, with collaboration with Tiziana Lembo.
But when you deposit very little virus on the LFD, as shown on the first slide, we are able to recover live virus if you eluted it in culture medium. So
that's
it's questionable.
Yeah, but that's cell culture supernatant. We do not see this observation when we use real clinical samples, which is what we will be shipping from endemic countries.
Just if I can, maybe we'll leave this discussion at the end of the session, but I have a question, a general question. Do you accept that I send you positive LFD without inactivation and send it not under UN 2373 or 2900 to Pirbright? No.
No,
okay.
we
So
you're not, but the point is, irrespective of what inactivation process you use, you're never going to be able to send those in the post because they contain full-length RNA. So even if you can prove consistently that you can't recover live virus from those LFDs, you will never be able to put them in the post. According to, UK regulation, I'm sure it's the same everywhere else, is anything with full-length RNA on it is considered to be a specified pathogen and subject to shipping. So you will always have to ship these materials Category B, and Category B allows live virus. So I'm saying, why would you bother inactivating the samples?
Don, I don't, sorry, Chairman, you're referring to UK regulations. I think it's actually quite difficult to find within the IATA regulations and the shipment of dangerous pathogens where it actually refers to there being a danger relating to non-infectious to humans or animals at this stage. I don't think it's quite as clear.
Actually, it is clear. This is IATA regulations, International Air Transport Associations, that cover UN 2900 and 3373. That is how it is described. 2900 is anything that is tissue culture. 3373 is potentially infectious and diagnostics.
Yeah, but those are obviously also guided by committees of people, including committees that can be set up from this room, as to the actual risk. Yeah, they can be. They're not set up by some other, higher authority. And there are people from representing OIE. OIE represents the international, let's say, interest in animal health on those committees. So obviously it has to be risk-based, but these things are not completely set, as is my argument.
Thank you, Keith. So we propose to move to the next speaker. Thank you. The next speaker is Dr. Fuller. She will give a talk on